Hi, my name is Dan Fu. I'm Assistant Professor of Chemistry at the University of Washington, Seattle. I was fortunate and honored to be one of the Beckman Young Investigator awardees in 2017 for my work on quantitative single-cell phenotypic imaging of multicellular systems. As a chemist and inventor, Dr. Arnold Beckman has been a lifelong inspiration to me. My research has been heavily influenced by Dr. Beckman's revolutionary invention, the DU spectrophotometer. In my lab, we combine optics, electronics, and chemistry to build cutting-edge spectroscopy and microscopy instruments for visualizing and measuring biological cells. The fundamental principle is the same as the DU spectrophotometer, the Beer-Lambert law. The DU spectrophotometer measures the concentration of molecules in a cuvette based on their light absorption. The signal A is the amount of light absorbed, which is proportional to the absorption coefficient epsilon, cuvette length B, and concentration C. The critical question we ask ourselves is how we can measure the concentration of biomolecules in a living cell. A cell is a lot smaller than a cuvette, about 10 micrometers in diameter. So signal will be 3 orders of magnitude weaker. Moreover, light is not only absorbed, but also scattered in all directions, making it challenging to quantify light loss due to only absorption. To address this challenge, we use nonlinear optical spectroscopy. Let's use red blood cell as an example. If we focus a near-infrared laser into the cell, nonlinear absorption will confine the signal to the small laser focus, about half a micron in diameter. This is because hemoglobin, the protein that carries oxygen in red blood cells, can absorb 2 photons simultaneously and move from ground state to excited state. This process ensures that the absorption signal we measure is only from hemoglobin molecules within the tiny laser focus that has a volume of about 0.1 femtoliter or 10 to the minus 16 liter. Using ultra-short laser pulses to increase efficiency and rotating mirrors to raster scan the laser beam in 3 dimensions, we can map hemoglobin distribution at sub-micrometer resolution inside red blood cells. With the new lasers and microscopes we built in the lab, we can now image tiny blood vessels called capillaries in a live mouse brain, measure blood flow and oxygen delivery, and identify abnormalities such as mini-strokes of the capillary. Using similar chemistry and optics principles, we can measure many other properties of living cells in the body. A molecule has many chemical bonds. These bonds have characteristic energies. When a laser is shining on a molecule, they can excite the vibrations of these chemical bonds. These vibrations give rise to the Raman spectrum, a fingerprint that can uniquely identify the molecule. We developed a technique called stimulated Raman scattering, or SRS microscopy, to rapidly map the concentration of molecules inside cells using their characteristic Raman vibrations. We can then measure the biophysical properties of cells that are otherwise difficult or impossible to measure, such as shape, mass, density, and growth. These properties are phenotypes of a cell and reflect the health and function of the cell. So why should we care about single-cell phenotypes? The human body has 37 trillion cells. The brain alone has 86 billion cells. Every cell is unique. In biology, we learned that a cell looks like a sphere. In the lab, cells are often taken out of their tissue environment and grown in a dish. And we may be more used to see cells like this. But in the body, cells come in many different phenotypes and perform widely different functions. Cells growing in the dish often behave differently from cells in the body. To fully understand the body function and disease development, we need to study cells in their native environment, know the identity of each cell, see what each cell is doing, and find out why they do what they do. The instrument we are building enables us to observe the cell unlabeled, unperturbed, and within their tissue environment. The phenotypes we are measuring allow us to classify cell types, cell states, cell growth, and even identify subcellular organelles with the help of machine learning. These advances have already enabled us to peek into the brain of a living mouse and start constructing a more detailed brain picture. In the future, we hope to build a complete catalog of cell phenotypes for detecting, identifying, and tracking every single cell in the brain and other organs, and monitoring cell interactions to better understand tissue physiology and the origin of many diseases such as cancer. I'm extremely grateful to the generous support of the Arnold and Mabel Beckman Foundation. They helped launch a new direction for my lab that focuses on building instruments for single-cell phenotypic imaging of living biological systems. I deeply appreciate Dr. Beckman's vision in fostering the invention of methods, instruments, and materials that open up new avenues of research and application in the field of chemistry and life sciences. And I'm proud to pass on the legacy from Dr. Beckman and train the next generation of scientists that will continue to invent better instruments and do cutting-edge science.